Okay, this is going to be the enzyme lab. We're going to do it in a few different pieces, so there's going to be a few videos to this. You've got an answer sheet that as we go through, you're going to be filling out. It describes what the enzyme is. I'll get to that in a minute. If we were in class, you'd be following a whole bunch of instructions. Well, you are in class, except we're in a group, and somehow the group has decided to let me do all the work. So you're my lab partner, but all you're doing is watching, and you're a useless lab partner. At least you could tell me when I'm doing something wrong. That's really what lab partners do a lot. Okay, the first thing we're going to do is set up how this is going to work. The enzyme that we're going to be using is called catalase. A lot of metabolic processes in cells produce hydrogen peroxide, which is potentially toxic. And what catalase does is it frees one of the oxygens off of hydrogen peroxide, which gives you oxygen paired together as O2, so not even oxygen radicals, and water. So that's a good product from hydrogen peroxide. So what we're going to do is we're going to make up a solution from liver. Liver's full of this enzyme, and so we'll have a solution that's full of this enzyme. So over here, we have liver, 50 cents off. And we're going to weigh it out. We've got a little scale here. If you come up here for a second. Okay. Turn the scale on. Not all scales work exactly the same way. Okay, that's a little weird. It doesn't seem all right. We don't want to include the paper when we weigh, so you hit a button that says tare, and it, come on, you can do it. It now will ignore the weight of whatever sort of paper or platform you're using to weigh out your stuff. Now, I need to open this. This is going to be a little juicy, so I'm protecting the table. And got to weigh out 10 grams of it. As we go along, I have to make sure that I'm following the instructions properly. Okay. Oh, big old chunk of liver. Got a knife. That is way more than 10 grams. Oh, this is going to come apart really easily. You may be picking up the bubbling of the boiling water that we're going to be using in the first set of tests for temperature. Let's see what we got. So we're about 4 grams. So that gives me an idea how much 10 grams is going to look like. There we're up to. Okay, I should have brought a pair of forceps over. Trim this down. It doesn't actually matter that this measurement doesn't have to be incredibly precise. If you started with a little bit more, it wouldn't really be that big a deal. I'll talk about that later as we do the prep. Let's see how that works. Excuse me. It's biology lab, it's going to be messy. Sometimes gross. Okay, that's weird. It weighs more than it did before I took that piece off. I have a pair of scissors. And now it's lost a lot more weight than that makes any sense of. Okay, 10.05 is going to be close enough. Now I need to stuff the liver back in the bag. Put the bag back in the container bag. Sometimes this is like working in a kitchen, is that sometimes you stick stuff aside and clean it up later, and sometimes you clean stuff up as you go along. Considering the mess this is making, I think we're gonna clean this mess up as we go along. Get the juice off the table, put that over there. Okay, we're gonna make a solution of this. We have a saline solution, salt basically, that mimics the osmolarity of tissue, because liver is tissue. And we want 50 milliliters. A little bit more. Okay, 50 milliliters, this is a 50 milliliter thing. I just realized I don't need to manipulate that with my fingers, so I'm going to wash my hands for a second. Okay, so put the cap back on that. Put a little bit of this into the mortar. This goes, whoops. This goes into the solution. Actually, it's good that we went a little over because some of the juice that's still in the paper has some weight. Ideally, I should have used a waxed paper that didn't pick up the juice, but as I said, precision not as critical in the ways here. So, I'm gonna mix this together and then I'm gonna grind the living daylights out of it. It does not want to be ground. It wants to jump out from underneath. Okay, I'm going to cut that up a little bit. You're basically squeezing the enzymes out of the liver. So the more squeezing you get, the more pulverizing you get, the better off it works. I'm almost thinking I have a different mortar. See if this one works better. This one does not work any better. In fact, that one works worse. What you wind up with is a solution with enzyme in it, but except for the fact that you measured out the liver, there's no uniformity to how much enzyme each, if we're working in a bunch of different groups in the lab, everybody winds up with different amounts and different concentrations of enzyme. That will relate to one of the questions you get at the end of the lab. But one important clue to answering that question is that you got to make enough stuff that you can do all of your tests with your starting prep. Because if you have to make a different prep, all of your results kind of fly out the window. To chop some stuff up. The large pieces are just slipping out from under the mortar, and the little pieces may do that too, but figure why not give it a try. Been a very long time since I did this initial prep myself. Add a little bit more juice. You don't want to add it all because you're going to use some of that juice to rinse this container out. It does work better in very small bits though. We're getting a much better grind on these things. The more enzyme you can extract, the better your reaction will be, the easier it'll be to get your results, to see what's going on, which is a really big part of this lab. Incidentally, the first time I set this up for a group of students, I set up the saline solution wrong, and it denatured the enzyme. So nothing worked. Took me a while to figure out what had happened. I just had mismeasured the salt going into the saline solution. Okay, next step is over here. We're gonna filter the juice from the gunk that's left over. So we have a filter funnel and filter paper. A piece of filter paper goes into the filter funnel. It's weird how the paper is a little bit bigger than it should be. Not entirely sure why, but that's always been true. And, I feel like I'm not quite ground up enough. Now that I got it up here looking. I'm going to start by decanting off some of the real juice. That'll go through the paper really easily. I'm going to do a little bit more grinding of what's left. This is the thing about doing experiments. A lot of times, the most important part isn't the test. It's getting everything set up so the test will run well. Okay. I'm going to let that keep going. Takes a while. You can see it's dripping down in there. In the meantime, we're going to be running tests with hydrogen peroxide, just the stuff that you can get from the supermarket. That's where I got mine. It is sensitive to light, which is why they put it in an opaque bottle. Let's see. Just to give you an idea of what we're going to do. I'm going to put a very small amount of this juice in the bottom of a test tube. And with a different dropper, because I don't want to put the contaminated dropper into the peroxide source. Okay, come up close. With my dark shirt behind it, that should be useful. You'll see what happens when the peroxide hits the thing. Releasing oxygen makes a bubble. And what we'll be doing in most of the tests is watching this column of bubbles rise up to a line, and we will time the rise until it hits the line. Now, I didn't put enough in here to get a really big reaction. See, if I put the rest of the dropper in, this is what you get. And so that's basically how we're running the experiment. Right over there. Another thing that's important, and it is part of the instructions, these are milliliter droppers. Well, that one is not very easy to read. that's way better. So there's a mark here for 1 milliliter. Whether you may not be able to read it, but if you were in person, you could read it. And a lot of times, that's what we have to administer. We have to administer 1 milliliter of stuff out of the dropper. So one of the things it tells you to do is practice. Okay, I had to squirt a little bit to get the level up. Now I've got a little bit of air in the bottom anyway. Tap a drop out. And I've lined the top of the water column up with that 1 meter mark. So now I have a dropper that's going to deliver 1 milliliter of liquid. And sometimes that takes a little bit of practice to get it so that you can do it reliably. Okay, we're gonna let this thing drain for a bit. I'm eventually gonna rinse a bunch of it off with the liquid, but I think this was our prep setup. This will work as well, getting ready for the first test, which is gonna be temperature, which is why we have boiling water and ice water and room temperature water. And in another room, I have body temperature water in an incubator. And we're gonna run tests. So while you're getting ready for that, take a look at your answer sheet and read through the whole paragraph and make a prediction of what you think is going to happen at these different temperatures. Keeping in mind that this is liver. I think it's cow liver, it might be pig liver, but the body temperature of a cow or a pig is very, very similar to ours. And knowing what you know about enzymes and optimum temperature and all that stuff, make a prediction as to what sort of comparisons you're going to get in terms of what should work well, what should work worse, what should not work at all, that sort of thing. Where you're going to be putting it in at body temperature, at room temperature, which is quite a bit lower, at ice temperature, which is really lower, and with boiling water, which is way, way hot. So make those predictions, and we will come back and do the temperature tests on the next video. That's it.